So we have one more talk for this session. So it's going to be Dr. Tim Peterson. He is a faculty in the School of Medicine at WashU. He co-founded VitaDAO and is also the co-founder of HealthSpan Technologies, which is developing mRNA lipid nanoparticle therapies, and BioIO, which is developing small molecule therapies. Please welcome him to the stage.
Hi, everyone. I'll go kind of fast so we can get to lunch. So yeah, I'm just going to, hopefully in my talk, I'll be able to tell you all about why we think infectious disease is important for aging. So, and I'm a scientist, so it'll probably get it a little bit, maybe into some kind of gory mechanistic details, but hopefully that'll be interesting to you. Okay. So just kind of an outline of what I'm going to talk about today. So I'm just going to give a background on our kind of approach, how we kind of got to the biology we're interested in. And we've kind of labeled this approach mechanism of action technologies, and ultimately it involves a genetic screening approach. And I know everyone has their biases about what's the best kind of way to do science, but I'm a big believer in genetics, and hopefully the research will help explain why we think genetics is such a powerful way to get to drug mechanisms and how to make better therapies. And so the meat of the talk is really around this class of drugs that we call CADs, cationic amphiphils, and sphingolipids, which are the metabolism they target. And I'll then talk about how we went from there to talk about how pathogens and infectious agents are important, and then ultimately how that connects up with aging. And then lastly, of course, why does this matter in the big picture for life expectancy in the world? Okay, so this kind of a platform we called a moat, mechanism of action technology. So everyone wants to have a moat for the drugs developing, and so we have an approach to do this. So ultimately, the work stems from not only our work, but for several other people that when you're talking about small molecules, I think one of the very surprising things that's emerged, mainly through genetic approaches involving CRISPR and other approaches, is that many small molecules don't really work for the reasons that people claim they work for. Okay, so we think you have a calcium channel blocker. Well, it's not actually a calcium channel blocker, or it's a calcium channel blocker plus does 10 other things. And this has come, and really this comes from the fact that most things, it really comes from the rational drug design school of thought, which has permeated pharma over the last many years. And it starts with the biochemical approach. You start with kind of a single target, you optimize around that target, but then you don't really consider what else in the genome, what else in the proteome that drug may be doing. You have something you think is highly selective for that one thing, well, but you didn't really screen everything. So, right, and that really wasn't possible until genetic approaches such as CRISPR came along and allowed you to survey the entire genome and really say, okay, if we did an unbiased survey, you threw your fishing rod in the ocean of the cell, what does it really fish out? And so that's essentially what our CRISPR screening approach allows. And so, like I said, there's many other people that have been on this idea or kind of doing variations. So I owe huge thanks to people like Jonathan Weissman, who's now at MIT, formerly UCSF, and Jason Seltzer has this very nice paper in Science Translational Medicine that gets to some of these topics in oncology. Okay, so just a basic idea of the screening approach, if you haven't seen this type of approach. So it is pretty basic. It may look kind of, there's a lot of information up here, but ultimately you just mutagenize cells. So you're mutating every gene in the genome in a single cell format, and you come in with a drug. In this case, I'm going to talk a little bit about alendronate, which is a pretty interesting drug for osteoporosis, and ultimately gets kind of volcano plot of results. So each one of those dots is a single gene, and you kind of look for your kind of outlier hits that are, in terms of p-value and significance for the effect. So we did this with bisphosphonates, and really I'll just skip through this real quick, but essentially what's really cool about this approach is that amongst the hits, you get kind of this bullseye effect where the hits flip from gene inactivations, which cause the cells to grow better, to those that cause the cells to grow worse. And you get this kind of bullseye effect as kind of shown by this kind of red, blue, red pattern where essentially you can kind of genetically identify the spot where the drug is acting. And so in pretty much every case we've done, we've actually found the physical target for the drug. So you're not even doing an approach that would you're not demonstrating the physical interaction, but you're basically, through the genetic map that you identify, you can basically pinpoint where the drug physically acts inside the cell. And so we went on to basically show, okay, so these drugs target, they're drugs that have 2-phosphates in them, and they ultimately target things which have some chemical mimicry with those 2-phosphates. So farnesyl diphosphate synthase is the 2-phosphate containing molecule or protein that people knew that existed, but there's several others, including this SLC37A3 transporter and another gene, atrate, which is a cofactor for that. And so we published several papers on this, including a Cell paper which was a little more indirect in how the cell ties together. But a lot of really interesting work came up from this kind of relatively boring drug, which no one really cared about. And I think we made some pretty interesting discoveries on how this works. And actually, it's still pretty much, to my knowledge, really the only validated, really pure gerotherapy in the sense that this drug, yes, it's used for osteoporosis, but it actually is one of the only drugs actually shown to have a mortality effect that's independent of its effect on bone density. So it's really pretty much, I know we talk about rapamycin and metformin and all these other drugs, but to my knowledge, bisphosphonates are really pretty much the only really validated therapy that exists today. Okay, so another drug we applied this to were antidepressants. So there's been a lot of literature on how antidepressants may not work on serotonin, so we were interested in this topic, and there's a bunch of other really well-known drugs that we've also studied that we're interested in applying our techniques to. So we took, sorry, the pointer doesn't work, but essentially several of these drugs up here are popular antidepressants. Sertraline is Zoloft, fluoxetine is Prozac, nortriptyline is another popular one. So we applied a screening approach to several really popular drugs that people all around the world are taking, millions of them. And essentially, what the really interesting thing that emerged from this is really a highly significant pattern that enriched for sphingolipid metabolism. So you can see the p-values are quite large, on the orders of I think 10 to the minus 30, some astronomically small number. And effect sizes, these were genes that scored basically in every screen we did. So very significant pattern, very selective, no really, pure signal, no noise essentially from these genetic screens for these widely divergent drugs that we're calling CAD. That I'll explain more in a minute on that they're CAD drugs. So essentially, yeah, like I said, when we surveyed these 6 drugs versus a whole panel of other CRISPR screens that we've done on a variety of other drugs, essentially, really enriched for the sphingolipid metabolism, and this metabolism is really not seen with any of these other drugs. And so, what are these CAD drugs? So, they're also known as lysosomotropic. There's been some work on these drugs, but the argument essentially is the effect on sphingolipid metabolism, the effect on lipid metabolism is really not relevant, and our argument is that this is actually the highly relevant mechanism by which these drugs work. So the key, in terms of the chemistry, to allude to the title of this talk, the key really take-home from this point is that these drugs all have these 2 specific properties. So they have a high C-log P, so they're essentially more hydrophobic. A lot of these CAD drugs are neuropsychoactive, and so they allow that hydrophobic part allows them to cross the blood-brain barrier well, so that makes sense. They're weak bases, and so they tend to get trapped in acidic organelles like the late endosome and lysosome, and that trapping allows them to actually be good drugs because they kind of persist long. And that's, I think why particularly the antidepressants take several weeks to work. So they kind of take several weeks to accumulate to levels which allow them to have their effect. So here's kind of chemically, this is just kind of how they look. It's probably hard to make heads or tails from these. You can obviously see more, phenyl rings. And anyway, the bottom line is these drugs have a high C-log P, high pKa, and they're distinguished in that characteristic. So it's not only antidepressants, but also actually hydroxychloroquine, which of course was kind of that famous drug for that ratcheted up for COVID, and I'll go more into that when we can talk about pathogens. But there's several pretty high interesting drugs in this collection. So the take home from this kind of part one here is that there are several drugs which share the chemical properties which allow them to inhibit the rate limiting step in sphingolipid metabolism. And this allows us to basically, our screening approach in general allows us to understand mechanisms of action of drugs. And in particular, I think we and others are really part of a wave of people that start to think, in particular, like for example, that the antidepressants don't actually work through their effects on serotonin, rather, and we argue it's through their effects on sphingolipid metabolism. Okay, so how does this relate to pathogens? So I was very fortunate when I was at MIT to work alongside Tyne Brumelkamp and John Corrette, who had really been pioneers in genetic screening, and they first applied this to a variety of pathogens. So several of these really nice PubMed indicated papers here are several Nature papers and very, very interesting papers by them. So we did essentially a similar meta-analysis. In the first case, we kind of said, okay, what's the common genes between all these kind of random small molecule drugs? In this case, we basically took all the screens by Jan and a variety of other people and said, okay, if you're talking about bacteria, viruses, toxins, parasites, what are the common human host genes that emerge from those screens? Very surprisingly, was that essentially it's the exact same genes that we found screening with these CAD drugs. So all enriched in sphingolipid metabolism. You can see this a little more clearly here. Essentially, again, if you take drugs, the drugs on the left are all non-CAD drugs. Drugs on the right are all CAD drugs. Essentially, as it's shown here, it's essentially an 80% overlap of the CAD drug and pathogen drugs. So suggesting that that if you use, suggesting that there's a strong genetic relationship between sphingolipid metabolism and the sphingolipid metabolism being the metabolism that's really critical for how pathogens gain access to our cells and inflict their damage in general. So, okay, so the summary for part two here is that, as I just said, sphingolipid-related genes are targeted by CAD and are common host factors importance to diverse pathogens. And then, of course, that begs the question, well, shouldn't we be using these CAD drugs for infectious disease? And really this idea is not, surprisingly, not really taken hold. There's definitely several kind of anecdotal pieces of evidence in the literature, but to my knowledge, it really hasn't taken hold. Very fortunately, a colleague at WashU, actually a psychiatrist at WashU, kind of leveraged our data. We have been collaborating on another project, and he took the CAD drug fluvoxamine, which is another antidepressant, and to my knowledge, this is the only repurposed drug that has shown promise for COVID. I mean, there's been a lot of other studies, but this is the only one that's been kind of validated by several groups. So I was pretty excited to be part of the work that led to that discovery. And they published a paper in JAMA 2 years ago on this. Okay, so how does this connect all to aging? So, all right, so we did this screen, we kind of mechanistically understand how these drugs work, but of course, what is the kind of downstream effect of inhibiting sphingolipid metabolism, and why is that relevant to human health? Okay, so the downstream effect, so you can take other approaches. So we just took a transcriptomic approach, again, a meta-analysis approach. So on the left, essentially, is the common gene expression signatures when you treat cells with CAD drugs. And on the right is the common gene expression signatures you see when you harvest tissues from animal. We took tons of different species and tissues from that had been infected with various pathogens. So very kind of diverse collections of experiments to find essentially the common signatures. And again, there's a very high overlap between the two data sets using CADs or the pathogens, centering on senescence and, of course, the sphingolipid metabolism that the CAD drugs target. So that you can find all your friends here, P21, SERPINE1 or PI1. There's a variety of other friends here, but essentially the signature that stood out was a senescence signature. And then, so what that implies is that essentially, if you use these CAD drugs, potentially one way they work is by basically inhibiting senescent cells. And we've done several experiments. This is just a very simple snapshot. But in the results here are just very, very binary. So CAD drugs, highly selective for targeting senescent cells, really no effect on proliferating cells. As you can see, kind of comparing the black to the red here. And then where you take a variety of other non-CAD drugs, essentially there's no kind of differential effect between the senescent and proliferating cells. So really, these CAD drugs are very highly, as the title says up there, they're highly potent, highly selective senolytics. Okay, so summary part 3. So I think really in terms of the chemistry, what's critical here is that this very simple chemistry, this high C log P, high pKa, it really predicts senolytic activity. So if you just have those two properties, so you don't even need to do a screen, right? You just say, does this drug have these two properties? And then if they do, it's a senolytic. So to me, that's a pretty impactful statement to be able to make. Of course, to be widely generalizable, we've got to do many experiments, and hopefully you guys can do some as well. But the simple idea is there. Just go look, find those two properties on PubChem, and that's a senolytic. And of course, it would work not just for pathogens. We actually did DNA damage in this case, right? Because ultimately the signature is a senescent signature can crop up. The idea is generalizable. It's not really specific to pathogens, but I think we found that it works there quite well. Okay, so in terms of the big picture, so so you probably all can appreciate infectious disease matters. Of course, COVID led to millions of deaths and actually a drop in the life expectancy, particularly in the U.S. But I'd argue that we just really haven't been thinking. We think of all these pathways and we've got all these kind of new approaches, but ultimately the biggest life expectancy gains, right? This is the kind of famous Preston curves up here, the relation between life expectancy and GDP. So just the future of our world, right, depends upon these kind of actually pretty simple innovations, right? Hygiene, sanitation, then antibiotics, then vaccines. These were the reasons that these curves shifted up, right? But we've kind of stalled out now, so we obviously have to approach, hopefully this is where our new approaches will come in. So, but I think the relationship is pretty clear that, probably infectious disease were key to all these major upswifts and not, of course we didn't have any of these fancy approaches we're coming up with now. And then just maybe another point to kind of drive this home. So this is our analysis of UK Biobank. So again, another meta-analysis. We took basically all the top aging-related disease, the lists you can see down there on the bottom. We said, okay, what are the common genes from this list? So you can see, of course, the ApoE cluster, which is a very famous, longevity gene, the insulin receptor. So these are two genes, two pathways that, of course, everyone appreciates is important to longevity. But really, amongst this list, you can see in these green dots here that these are genes important to the pathogen host response to a variety of pathogens. So if herpes, for example, a very kind of underappreciated pathogen, this infects 2/3 of the world's population. So the fact that its receptor is a key longevity gene should probably not be that surprising. And then, of course, there's been a body of literature which has largely been ignored that actually herpes is a major cause of Alzheimer's and other age-related dementias. So I think more work needs to be done in this area. So, okay, so just thank a lot of people that have contributed to the work. So the team at BioIO, various collaborators at WashU and other universities, and then funding sources. And VitaDAO is an organization I've been part of, and they're funding this work in particular. So thanks for your attention.
So thank you so much for that. And I'm actually not surprised that the psych medications have a different mechanism of action at all. So we've known for a while that that these serotonin reuptake inhibitors, that by the time they start working, which is 2 weeks in, that serotonin levels are back down to baseline.
Exactly.
So they definitely don't work that way. So I'm not surprised to see this at all. But this is really cool.
Thanks. Yeah, thanks.
Yeah. Is there maybe one or two questions?
Yeah, just a big picture question. So the CAD structure itself is very simple chemically.
Yep.
So actually very potentially different chemical structures.
Yep.
How is that possible that you have aging senolytics, infectious disease and
Yeah, I mean, it just underscores really how important sphingolipids are. So these enzymes chemically mimic sphingolipids, which are also essentially cationic amphiphils. So sphingolipids are just massively important. So for example, that right there, they are one of the two main components of lipid rafts, where essentially all cell signaling, all pathogens gain access to human cells. Not all, I mean, but the vast majority of them have receptors that are sitting in these lipid rafts, which are highly sphingolipid enriched. So they're a key node on the order of cholesterol or insulin signaling, just kind of underappreciated as such. In fact, the original naming of sphingolipids, right, is like these were lipids that were in the brain. I mean, I don't know what a sphinx is, but essentially the argument is these were kind of mysterious lipids that are in the brain, highly enriched in the brain, that people just, the original person who identified them in the 1800s couldn't understand what they were. So they just kind of just been less popular than other metabolism.
Do you know of any kind of like natural products that have those similar properties?
Yeah, I mean, definitely there's several. I mean, yeah, there's several. So myriocin is a drug, is a small molecule that targets that same enzyme. So yeah, they're just. I think again, it's just kind of an underappreciated area. I think if people looked at it more, they would. There is a big sphingolipid community, but not as much as you would think. So yeah, other metabolisms just kind of outpaced it. Yep.